I'm Jen Wilson from UCLA, so I work just upstairs. Thank you to the organizers for inviting me. I'm not traditionally a geneticist and have no formal training in genetics. I'm more of a systems biologist with some computational work. But I always find these collaborative meetings exciting because sometimes when you come to a field with like either too much ignorance, which we'll get to, and you can decide, or just like a different way of thinking, this is where you can come up with new and exciting collaborations. So the title of my talk is Predicting New Drug Phenotypes by Simulating Off-Target Effects. So again, we're sort of in the drug and therapeutic space. I'll tell you a little bit more about what I mean by that, but like if you already start to think about like drug repurposing, like that still puts you in the right sort of frame of thinking, but I'd like to add a little bit of nuance and detail to that. So one of the things that you might appreciate, and starting from a very simple idea, is just that we already know that medicines can affect multiple outcomes in patients, just from a simple idea. Anyone who's seen a drug commercial, I listened to my uncle ramble at Thanksgiving that, you know, the drug does something cool, and then there's that really long section of all the side effects. And so I always like to pick on metformin. It's an anti-diabetic that's been widely prescribed for decades, and it does treat diabetes. It also has various side effects like nausea or diarrhea. And there's many other things that have been sort of associated with long-term exposure to metformin, one of them being improved outcomes in oncology, for instance. One of the other things that we also know is that patients also experience multiple different outcomes or phenotypes, if you will. And so here's my little cartoon, and one of the things that I'll be leading into is that like now that we have large-scale electronic health records, it's easier to track and essentially compute over this data. But in the little cartoon, there's patients here. There's a bunch of orange triangles, so in my talk, drugs are triangles, and I'll remind you of that as you see figures. And then the little boxes here are the different outcomes that a patient might experience. So there could be comorbid conditions, so this patient has depression and diabetes. And then the yellow ones are sort of like side effects that they might also experience. And so just like with the sort of understanding of the drug, we see this in the health record. But another really interesting thing is that like if a patient has diabetes or depression, they actually see different specialists for each of those. And so from a systems biology perspective, you sort of look at the healthcare system and you say like the way that patients deal with any of these individual conditions is like by highly specialized interactions with, again, like an endocrinologist or a psychiatrist, for instance. So the way I sort of pitch the lab is that we can think about what medicines do by how they interact with the cell. And so I'll go to do just some basic molecular biology. But the idea here is that the cell is sort of like a bag of proteins. There's lots of different databases of how proteins interact. And the way that we think about medicines and drugs is that they traditionally bind a protein. And often the development space kind of thinks about these as like there's a drug target, like the drug, if you've been following Alzheimer's, like binds the amyloid plaque, or there's many oncology drugs that bind a specific receptor. But actually, one of the ways that they exert their effect on the cell is through this network of protein interactions. The same network, not surprisingly, is the one that also gives rise to side effects. And so sometimes, as this cartoon kind of suggests, is there's sort of like a parallel pathway for efficacy and safety, so you might be able to manage both effectively. But a lot of times, they're sort of intertangled, and that the side effect is sort of a byproduct of the on-target or intended effect. So again, as a systems biologist, we can now model these things. We can model protein interactions and how drugs change the cell and have relationships to both genes and proteins that are associated with what the disease or what the drug is intended to treat, as well as some of the side effect phenotypes. And then if you think back to our patients in the health record who are then, you know, experiencing multiple conditions, and then they have all these different things that could be relatable in a network, the healthcare system is not necessarily set up to think about those things systematically. So I sort of pitched the lab as saying, well, you know, we can model these, and maybe if we have more robust models, we can better treat patients or personalize their therapy based on the spectrum of their own conditions and which things you might want to treat. So there might be one antidepressant that could manage diabetes and hypertension, for instance, in some extreme example. So then what I started doing as a postdoc is that I built this algorithm just to sort of like play test that idea and quantify where we're good or not good at this. And so not surprisingly to this audience is that you can build models that will simulate multiple effects for drugs. And so again, drugs here are orange triangles. They bind red circles, which are drug binding proteins that we get from different data sources. And then the thing we add is we do a little bit of a pathway search, which I'm happy to talk about. I don't have the math here, but we sort of collect the highest quality downstream proteins around a set of drug targets. And then that's sort of the draft pathway. And we do that searching per target, and then take a union, and you get a union of protein-protein interactions that we think are the drug's pathway. And then we just do a statistical enrichment, nothing fancy here, looking across multiple databases to say this network's enriched for diabetes, oncology, different phenotypes. And then essentially those become sort of the predictions of what the drug is meant to do. So it's a relatively small network, basically given the scale of computation available now. So R has about 300,000 edges across 25,000 genes, proteins, and we have some gene variants, which I'll get to eventually. And then interestingly, for 7,000 approved and experimental drugs, we predict around 1 million drug effects. So still sort of like not as big data can be, but this number of effects is many more orders of magnitude than what we sort of know to occur. So if you look at the actual health record, there's only about a couple thousand approved drugs, where often a drug is approved to treat one thing. Sometimes it's approved for more than one thing, but you get like tens of thousands of sort of known validated drug causes disease or drug causes side effect. But models of these scale can predict millions of things. So how do you use them wisely if you don't know that 900,000 of them are sort of unverified? So that was one pitch here, is just that you don't have really good standards for them. So again, I'm going to pick on metformin just to show you a real network and make one or two other points. But sort of at the crux of what we do is some form of making these circle line diagrams. So here's metformin, the anti-diabetic. I pick it literally just because it makes a smaller network and it fits on a slide. But we have 5 drug binding proteins in our protein interaction network. We add a whole bunch of gray circles, which are the downstream proteins, and then you'll see that there's some gene variants here, which I won't necessarily talk about now, but we can also sort of model genetic interactions. And then there's a whole bunch of green boxes that get added. So we make like 20 predictions for the effects of metformin. When I built this, it wasn't a traditional machine learning model in that it's not trained to know that diabetes or metformin treats diabetes. It was just using the amount and quality of data that's there. So behind all of these edges, there's a weighted interaction network that reflects our confidence that the two proteins actually interact. And then you can essentially do an empirical search of the network, again, because ours is relatively small, and say these are the highest quality proteins around these drug targets compared to the entire network. And so again, it's mostly sort of data-driven, and it wasn't sort of trained to know that diabetes was here, but things like diabetes occur downstream based on the quality of the evidence. And the reason for doing that as an engineer is because you say, well, now that I want to go build a model for Alzheimer's or something where there's not good training data of like this drug actually influences the disease in a favorable way, you want to have some confidence or some method to get you close enough to new targets or new pathways. So the whole pitch here was just that we can do this sort of modeling. It's not highly performant at the time, and so we did this benchmark. This is maybe 2016 now, so it's a little outdated. But for about 238 drugs, or again, 1,756 drug disease pairs, because of this subset of 238, some of them were approved for more than one indication or disease. There's some fraction that fit this paradigm, and then there's a whole lot that don't. So we have a lot to learn if we think that this sort of downstream modeling approach will, in fact, give us predictions that we have confidence in or that can lead us to better outcomes. But the theme here is that I was fortunate to spend in my postdoc some time both at the FDA and then spend time at Genentech. And a lot of what I saw is that most of drug development is sort of this paradigm on the right, where we have one triangle, one circle, one box. So there's like a mutated protein, the drug binds that protein, and that protein's associated with the disease, the green box. And sort of the idea here is just that like there's enough confidence in the data or the approach to then say some drugs are going to bind more than one protein. Maybe we should just embrace their off-target effects and the fact that they might also induce more than one thing. But if we can build these models more rigorously and in a way that we can test them reliably, then it sort of gives rise to this network-based drug development. Okay. One other thing I'll point out, I don't necessarily need the animations. I just sort of recolored this chart just to make one thematic point. So one of the other things that we do in the lab that allows us to sort of bridge between these like molecular simulations and then what's in the electronic health record is that we'll do the same modeling for all different types of drugs. We'll take them from any class or from any disease area. And this point is just to say that now if you have a drug that treats depression, treats epilepsy, or treats asthma, we sometimes find these sort of non-intuitive connections where you would think, depression and epilepsy are sort of neurological, asthma maybe a little bit less so, but for these drugs that are from different classes and different use cases actually end up sharing patterns in the network, and it's often downstream of their targets. So just to make this point before I go on, this is sort of one of the things that we do where we can now do these like non-traditional groupings of drugs and then go back and study them in the health record. Because a lot of times when people go back to look at the health record, they'll do a repurposing study of like all antidepressants or individual types of antidepressants for some outcome, but they don't necessarily include one antidepressant, one anti-epileptic, or one anti-asthmatic drug. Okay. So I sort of walked you through some of the protein interaction modeling stuff and the way that we approach problems. There's lots of things, obviously, to solve here. So There's lots of different questions, and I won't talk about all of them today, but again, to give you a flavor of like where the systems biology work in the lab goes. The metformin story is really nice, but one question you could ask is, what do all the other green boxes mean? In that, the other point I was trying to foreshadow for you is that we make many more predictions than there is evidence, and so we're trying to understand this idea of over-prediction, and some fraction of those things might be actual real things, but financially there's no reason to do another clinical trial because in the US you can take drugs off-label, and so, but you can like informatically gather evidence and say with more confidence that some of these other unverified predictions have evidence either in the health record or through other molecular biology data. The one I'll talk about today, which will, I think, hopefully lead to the most interesting discussion, is what's the utility of downstream proteins, which is to say something that's different about what we do compared to some of what you've seen with like graph neural networks is at the end of the day, as like more of a bioengineer, I'm sort of interested in like which proteins are we actually predicting. So like if something is downstream in the pathway, like what does that say about the biology of the disease, or why antidepressants or anti-asthmatics are similar. And then we do a lot of other things about how do we know that our data lists are correct. Of like, if you look across like 6 well-known drug target databases, they actually agree very little for like the same drug. So often there's one target that's been, one protein where the drug binds this protein and it's shown up in FDA review and like early development, but then there's a whole list of other predicted binding partners for the drug that just don't agree across databases. So I won't talk about that today. I'll sort of talk about the second story. And I'm going to tell it through sort of 3 different stories, and depending on time, I might do the risky thing and insert something that's a little bit more genetic focused, but very underdeveloped, just for the sake of like taking advantage of people's expertise. But I'll first give you a story about predicting drug-drug interactions, and then we did a little bit of this sort of new target discovery in ALS patients. Again, bridging what we predict in the models for approved non-neurological or non-ALS drugs, and then trying to bridge that to the health record. So when I spent a little time at the FDA, I learned a lot about drug regulation as well as this idea of drug-drug interactions. And so one of the things that I sort of misunderstood even when they started talking about drug-drug interactions, I assumed it meant in an interaction network, but really what they mean and the way that they regulate drugs that might interact with each other is that drug 1 and drug 2 often bind the same protein. And often it's a transporter or a metabolizing enzyme. And if you know that your drug is processed by a CYP enzyme, then patients who are on any other CYP enzyme drug sort of get predicted to be possible interactions. Not surprisingly, it's infeasible in any clinical trial to actually test the full range of drug combinations that a patient could experience. And so regulating the transporters and pharmacokinetic proteins of the drug is kind of their primary way of predicting or anticipating how 2 drugs might interact. Not surprisingly, the sort of misunderstanding I had was like, well, certainly if 2 drugs have unique target sets, but actually have targets that are in the same pathway, or like the proteins themselves interact in the network, you could also induce an adverse outcome. And so as I sort of foreshadowed, we can draw network models and then we can do this prediction. And then so what we did to start out was that we derived new protein classes. And again, what I was trying to foreshadow with that like anti-asthmatic and anti-epileptic drug is that you can say, here on the left side, there's a bunch of drug networks, 1, 2, and 3. They're all associated with sepsis, which is like a severe bloodborne infection. Drugs 4 and 5 are also associated with sepsis, and we can make that prediction in our network. And then what's in tiny shading here is this protein A, this like dark gray circle, which is just to say there's a pattern for these ones on the left where they have some downstream connection. Again, unique target sets, maybe unique drug use cases, and drugs 4 and 5 don't. And so we can derive these drug classes. They're typically bigger than 5 drugs, but again, it makes a nice cartoon. And then we essentially go about predicting other drugs, these are now blue triangles, that are not associated with sepsis on their drug labels. But in this case, I'll get my laser pointer too. So A2 binds A, but it also doesn't bind anything that drugs 1, 2, and 3 bind. And the little red circle here for drug A1 is to say, if drugs A1 and 2 already share one of these CYP or metabolizing enzymes, it gets excluded. And then since I was fortunate to work with the FDA, again, this is a little bit of an outdated data set, but at the time they had extracted and scrubbed all severe side effects from the drug labels. And so to give you a sense of what we're looking at, they're like sepsis, edema, pneumonia. We also look at myocardial infarction and pancreatitis, and there were about 34 of them. And then just to give you a sense of like, there's a wide range of drugs that are associated with these things on their labels. And for anyone who hasn't read a drug label, they're usually like 70 to 100 pages. They're quite long. They show all the clinical development, all the manufacturing and technical information for the products. And they have 3 sections. There's a warnings, box warnings, and precautions section. And they did some just like natural language processing and saying, you know, there was some evidence somewhere in the trial that triggered a regulatory action to say like, somewhere in the development of this drug, there was enough evidence that it made it to the status of a warning. So it wasn't just like one patient who had some other comorbid condition. It was something that was sort of sufficient to be associated. There's other data sources of side effects where people spontaneously report, but they're maybe a little bit less verified. So we could talk about drug side effect reporting if you're interested in it, but that's the data set that we used. And they had at the time about 1,900 drugs. So again, this was collected in 2017, it's a little old now, but just to go back to, again, we make millions of predictions. There's only even 2,000 drugs that were sort of approved at the time that had a label to study. So we first benchmarked our predictions on a data set 2 sides, which is derived from one of the spontaneous reporting systems. And there's this large funnel to show you the numbers, but the one that I'll get you to is that I'll make 2 points. Here in the middle is, again, to give you a scale of like just how many proteins have data, and for this is that we could predict about 19,000 drug-drug interactions. But then you filter that by like, there's some drug combinations that are never prescribed in the real world, and you're like, okay, we can't test those. And then we had to sort of get sufficient numbers to put them in our little drug classes. And so at the end of the day, we had 405 combinations that were prescribed in the real world, and then essentially had drugs with or without the protein in their class. And then some of those were associated with more than one interaction, so it's 457 drug-drug interactions. And then 308 of them had a report in 2 sides, so they also were verified in a spontaneous reporting database instead. So it gave us like about an 80.7% sensitivity, just again looking at like, do the drugs bind things that interact? Again, comparing for like different permutation tests. And so then this was the first opportunity to bridge to the health records. I'll give you simulated data first. This figure is the same as before, just sort of reoriented as a reminder of how drugs get grouped. And we did 2 analyses. First, just looking at the relative risk for an outcome between drugs 1, 2, or 3, or 4 and 5, because you know, if like one of them is just more risky for sepsis, it would be misleading. And then we look for co-exposures in the health record. So you can look at the start and end date of a prescription as a proxy for when a patient might be co-exposed. And then you say, now what's the relative risk of when they're co-exposed and have some overlapping exposure, and these are typically within like tens of days of exposure. And we just measured a hazard ratio using standard treatment comparator control statistics from epidemiology, and then look at the change in the heart hazard ratio, and for lack of anything more elegant, it's a hazard ratio, but just to look for shifts. And really as an engineer, the goal was to use this shift a little bit for drug-drug interaction discovery, but also to verify that, you know, this protein that's downstream of these drugs with different uses is sort of relevant to its function. So I'll show you a couple examples. The first one here is looking at pancreatitis, and there are 2 classes. And instead of being the A protein class, it's TEN, which is to say these networks all had the co-occurrence of 3 different proteins, so it's a little bit different. It's P53, EDNRA, and NF-kappa B. And this is for the outcome of pancreatitis. And you basically see the relative risk between the 2 classes, with or without these 3 proteins, is it's relatively less risky, and then it's slightly more risk with the combination. There's really wide error bars here, because once you start looking for co-exposures for patients and then separating so that they can't be in either drug cohort, they do get really small. So it suggested that like, okay, maybe there's something here, but yeah, not statistically significant, but interesting from an engineering perspective. And then this is more similar to the cartoon in that these are looking at drugs that are associated with sepsis. There were many more patients taking these drugs, so the error bars get a little bit smaller, but you basically see that there's relatively less risk if you just compare the A drugs to the non-A drugs, and then when you have the combination. So it just suggested that some of the proteins that we're using to describe drug function were in fact relevant. So we did this for a few other classes, and the punchline here is that you can kind of get a sense of how many other experiments we were able to conduct based on having enough patients to do them. And then the subtle change here is that we, again, saw some that were just like the 2 examples I showed you, where there was an increased risk, and then there was 1 or 2 examples where there was a protective effect. So we can also detect drugs that might like reduce the outcome of a side effect if taken in combination. So the big takeaway there is that we can do the systems modeling, we can bridge it to the health record, and then this like whole network classification of drugs might be useful for learning new things. And so that gave us some precedent to now study ALS, which is a disease that doesn't have a lot of treatment options. And so this is a really similar cartoon to what I've shown you before, except now we have an arbitrary drug 1 And now I've highlighted just a different protein. It's C1R. It's part of the complement system. It's part of the immune system. And then the one other point I'll make here is now the boxes get a little bit more specific. So the platform is very flexible. We can update it and have it associate with any different sort of data sets or pathways. And so this was a really cool opportunity to collaborate with Sanofi, and they had a bunch of patient-specific data, both proteomics, sequencing, and so we could build the pathway models not from just like generic definitions of diabetes or ALS, but like sequencing data or proteomics taken from patients. And then do the same sort of modeling and say, for approved drugs, do any of them have any like distant connections to these sequencing or proteomics data sets? And then just to be transparent, here's a real example. It gets more chaotic than the metformin example, but this is acetophenazine. Its network is 235 proteins. It's associated with 138 phenotypes, 4 of which I've selected here because they're associated to ALS as well as other neurological conditions. And then just to make a point here, there's 4 orange circles. So again, this is one pattern that we observed across many different drugs containing some of the complement system proteins. And I picked complement system for a reason that I'll explain later. But just to give you a sense of, again, what the drug pathways themselves look like. So I foreshadowed this well to try and have you thinking about it. But so here's an example of drugs that, again, get classified as these complement changing drugs. So we have a steroid, methylprednisolone, gabapentin, which is the anticonvulsant, and mirtazapine, which is the antidepressant. So I was trying to foreshadow some of the drug classes to show you the real ones that show up and are prescribed in the patients. And then just to make the same point, this is an abbreviation of their network, but they have distinct red circle names. So they have different connections in the network that converge on something to do with complement system, which is also associated with ALS as well as some of the other neurodegenerative diseases we looked at. And then just to give you a sense, there's drugs in different use cases that are also associated to the same pathways and same patient data, but don't have a connection through the complement system. And I just, again, wanted to make the point that we, again, look at non-traditional drug classes. So there's an anti-asthmatic here, a blood pressure medication, as well as some antibiotics. So then we did the same thing. We basically looked at patients with ALS who have blue drugs and patients with ALS who have gray drugs and looked at overall survival. And so this is just a Kaplan-Meier plot showing the survival time. And to make a few points, again, it's really subtle. The blue lines are these ones that are sort of on the bottom and the gray lines are on top. This is not a statistically significant difference. And this was measured in the nationwide Optum market clarity data set. So it was a pretty large cohort, which in ALS, it's typically really difficult to do any sort of drug repurposing studies because there are too few patients. But using a nationwide claims database, and then also because we grouped more drugs together, it allowed us to keep more of the drugs, or more of the patients in the study. The other point I'll make is this goes out to 10,000 days, like it's 30 years. It's a really long time. And so it basically shows that it seems like some of those blue drugs are associated with slightly worse survival, but in maybe single-digit years. And then in the long term, it sort of washes out. But certainly at this point, I've showed you a proportion. There's very few patients surviving with ALS that long. There might even be misdiagnosed. But it's important to, again, make that point about the duration and the time and the size of the effect. Because the interesting piece of context here is that most clinical trials, especially in these disease indications, are 6 months to a year. So they're very short, and they're often tied to like functional ratings, like are you better at like getting dressed in the morning? And these other things are more immediate indicators of symptoms because practically that's what you have to do, and that makes sense. But the difference in the observational studies is you can follow patients much longer, and you can follow to maybe terminal endpoints that you care more about. So we also repeated the analysis in myasthenia gravis patients. And again, we see kind of like a slight disadvantage. This one's slightly more significant because there are more patients. It's marginally significant, so it might still not be compelling, but I'll tell you why I picked it in a minute. There seems to be something very confusing happening in Parkinson's, so I won't talk about that now. But the point of starting with the complement system and myasthenia gravis is that outside of the data set that we used, complement inhibitors have been approved for this disease area. And so this is a publication this article is from July of 2024. You don't have to know the drug name, but I'll just tell you that it's an anti-C5 antibody. And it was approved, again, in October of 2023, so relatively recently. And our data snapshot ended in February. And we also went back and just double-checked. None of the patients are taking any actual complement inhibitor drugs, so that wouldn't have confounded the analysis. Again, the drug was approved on a trial that ran for 1 year, from May of 2022 to May of 2023. And again, the thing that they measured is this mean MG-ADL, which is one of these functional rating scores. And so kind of where we were sort of pitching the story and where we were trying to pick complement as an example is that, we could see that sort of in the data before the clinical trial, like without the clinical trial or without exposure to the actual drug, that it seemed like drugs that would change complement could make things worse for patients. And again, we saw that at early time points that are on the scale of a clinical trial, but we see that sort of wash away later. We also did some experimental work with collaborators, and the only takeaway here is that for some of the blue drugs, we saw induction of complement in a stem cell model. Some of the gray drugs also induced a little bit, and there's clearly a little bit of differences here, but it was just like enough of a hint to say that, again, our protein models don't predict like if a drug induces complement or blocks it. But then like modeling it, following them in the health record, and then doing separate experimental work sort of like verified that like one of the off-target effects of these drugs is that they change the immune response, and that we could also sort of associate that effect with overall survival. So if anything, we were kind of saying there's some information there that might allow you to predict which targets are viable. And then I tell that because we did the entire analysis for different protein classes and different proteins. And so that complement system one here, again, not the most significant in our study, but was again the one that was already like developed and seeming to have like short-term benefits from patients. And so if anything, you might be able to use these sort of systems biology models of off-target effects, looking at health record to then say some of these other chemokine and immune-related pathways, as well as some of the like neurosignaling might have a stronger effect or might also be useful or worth exploring. So we pitched it both as a biological discovery application as well as maybe like de-risking as you make choices to like move forward with a target in development. So, and again, for the bioengineering side of things of building models, it gave us more confidence in the things that our models are selecting. So I've talked through some of these, but again, we can group, drugs grouped by these downstream proteins had measurable changes in survival. We still pitched it as like an ability to learn disease biology directly from patients. And again, because the health records are large and more robust now, UCLA participates in a UC-wide health record. That's a combination of UCSF, UC Irvine, UCLA healthcare system. So we actually have access to about like 10 million patients. So it's usually a pretty good cohort size to do some of these different studies. And then for us, again, as engineers, it said sort of something about the pathways we were building might be relevant to the diseases that we're looking at. So I think I definitely have like 10 minutes. I won't go over. I think for the sake of today, I'll do the slightly risky thing of adding something new that's kind of young, like underdeveloped, because we're trying to now bridge into the genetic space. Let me go back to one cartoon first. So I've showed you a couple of these like cartoons that are just circles and lines connected. One of the things that this gives you the opportunity to do, and which we're starting to explore, is that the nice thing about the health records, we also have documented gene variants for patients. And so we can look at patterns where patients are taking drugs and there's like a variant downstream. And so we're still doing some of the analysis there. But one of the other ways that these models are sort of deficient and people have started studying this is that they're not directed. So like we went into this not knowing if a drug was going to improve the disease or make it worse, and sort of what the direction of effect was. So we don't know if you administer this drug, if it like blocks the complement system or induces it. And so what we decided to do and tried to look at I've added a question here, but how do you know if your pathway is relevant? And the way that people in the systems biology field get to this is what's called pathway orientation. And the way that they typically do it at big research centers is that they treat a lot of cells with different drugs and then measure gene expression or sequence them. And you have these in vitro assays where you can then say like, oh, there's patterns. All these drugs induce complement. So there's an edge that's an activating edge. But one of the things we tried to do instead is that we wanted to then look at some genetic data. And this is kind of where we sort of wandered into maybe the space of co-regulation as a way to kind of get at what's happening. And so what we did is that we went back and looked for subsets of our network, and I'll start you down here with the red circle. So we're going to call this a center gene and this a neighbor gene. And there's really good databases of EQTLs now, and so we started with DICE. Some people use GTEx. We just haven't got there. But the mapping from systems biology to perturbation genetics is that if you have an EQTL, often there'll be many different SNPs that are associated with changes in expression to the center gene A. We just asked a simple question of like, is the gene downstream, this neighbor gene, also sort of co-regulated? And this might occur because of like multiple hypothesis correction where the SNP is not directly associated statistically with the downstream gene. And so again, none of these actually have an edge between the SNP, but the question is sort of just like, do we see any patterns here? And so here's an example again of like one of our little circle diagrams. This is the ephrin receptor. It has multiple different variants and then there's multiple different downstream genes. And so this is our center gene, and then these are all neighbor genes, and we say, like, are any of these sort of co-regulated? Can we kind of get a direction of what the edges are? And I'll pick on YES1 here because I just have data to show kind of what we're looking at. There's 35 different variants that are associated with EphA4, and it's in T cells I'll, for whatever reason, have the slide afterwards, from immune cell types, but again, these are healthy patients. And then this edge weight is the beta value that's reported by the data source itself. So there's a EQTL effect on EphA4 of this magnitude. And then what we go back and do is then we take their raw sequencing expression and then just bin them and say, like, do you have the reference allele or do you have the alternate? And then just look at the differential expression in those columns. So we're just still like very much looking at the raw data. And so then if you go back and you just plot, like, for all patients, what's the co-expression of EphA4 and YES1, we see that they're not really well co-expressed. You also get a sense of like the RNA-seq counts for each of those genes. But then if we instead do the difference in the alternate minus the reference expression for that downstream, that YES1, and correlated, again, we're using edge weight here, it's the beta score from the EQTL, you start to see that they're slightly correlated. And then we also plotted it just because I wasn't sure, but I wanted to see the data. If you look at the difference in expression for the upstream EphA4 on the x-axis, and then that downstream on the y-axis, this YES1, you again see that they're correlated, but only if you bin patients by both having the reference and the alternate allele. This came from a data source, the DICE database, which looks like I don't actually have the slides, so I'm sorry about that. But it's, again, this hints at they did sequencing in healthy immune cell types. So there's about 91 patients. They have 11 different immune cell types. And so this particular interaction only showed up in one immune cell type. But we can do this across different immune cell types. And so here's another example where GPR18 also has multiple variants. It's a G protein coupled receptor. And now you can see that the example that we picked on happens in T helper cells. And this is a relationship between GPR18 and one of the adapter proteins. But again, you see that there's multiple different variants that influence GPR18's levels. And so again, this is just that beta value. And then just to show you the numbers, again, it's still only 91 healthy patients, but we still get more than like somewhat balanced cohorts where you can then look at the difference in expression of the patients who have the reference or have the alternate allele. And so now you see a really similar plot, except there's a lot of colors, because now you see that this showed up in multiple of the different immune cell types. And if you just look at these two genes and their co-expression without separating by variant status, it's kind of like a cloud. It's not very well correlated. But then you start to see a little bit of correlation if you look across all the cell types, again, breaking it down by patients with or without the variant, and then just looking at the difference in expression. So this is like one way that we're trying to bridge into the genetic space and think about ways to like orient our networks using sort of more of like a human scale perturbation. There's some long-term goals there to make that more actionable in terms of maybe like understanding disease biology, or we obviously want to repeat some of this in GTEx. But it suggests that like some proteins that have documented molecular interactions seem to have some sort of maybe co-regulation pattern with a very like loose definition of co-regulation. So I'll stop here just to respect time. I sort of talked to you a little bit about network protein mechanisms. I talk about bridging that to the electronic health record and maybe learning new biology as well as like understanding what happens in patients. And again, all the theme here is that we like do something downstream of drugs using protein interaction networks. And I didn't get to talk about this one today, just there wasn't enough time, that always happens. But those are sort of the themes of things. And then what I tried to end on is like we're trying to wade into the genetic space. I think there's a lot of ways to bridge what we do in systems biology and in clinical data with what happens in understanding big biobanks and resources. I also want to thank the lab. It's always humbling as a new person that people come to work with you and they do exceptional things, and you learn a lot from them. And also to thank our funding sources. Again, I've been fortunate to sort of bridge the like basic science, and then there's opportunities to collaborate with industry and see really cool patient data sets. And yeah, thank you for your time. I know it's early, so happy to take any questions.